My name is Chris Holder. I'm from Johns Hopkins University. I'm in charge of the lightning sessions for the graduate students. So if you've never been to a lightning session, it's a very rapid fire pace presentation. So each student will have 2 minutes and 15 seconds to present their research. So if you have a short attention span, good news. Unfortunately, I don't think anyone has any animations of Klingon ships getting blown up, but yeah, maybe somebody will speak in Klingon or something. All right. So first up is me. My research kind of addresses part of the question that people have been pointing to all day long, in that models produce differences in their output, but it's unclear why they do that. So my advisor actually came to me with this problem, and my other passion is machine learning and its applications. And so we'd been using machine learning to find relationships in different phytoplankton systems. And so we were curious whether we could apply this to more complex biogeochemical models as well. And so the first thing we did is we took 2 cases from the biogeochemistry model known as BLING. Basically, we have 2 different versions here. What you're seeing on the bottom is the control case, and then up here is just the same thing with higher CO2. So when you compare the outputs to each other, you can see that there's some similarities, but for the most part, they do have differences. So obviously, the first question we wanted to ask was, can neural networks reproduce the results from a biogeochemical model? And the answer is yes. So the predictions of the neural network for the pre-industrial control that we had versus the actual observations was 0.98. And when we did the same thing for that high CO2 case, we got another 0.98. So then we wanted to get into the question, can neural networks be used to show any sort of relationship within these simulations? And so what we did was we took a neural network, trained it on this pre-industrial case, and then gave it the inputs from this high CO2 case. And these are the observations. So the implication being there that that trained neural network was actually able to capture the underlying dynamics of that system. And then the real cool thing is we can do a sensitivity analysis where we are able to maintain variables at some constant value and then allow one of the variables to vary between its minimum and maximum. And so when we ran this on 3 cases, so here we have the pre-industrial control, a higher CO2, and a higher lateral mixing term. But basically, the neural network showed that every single model had the exact same relationship, which is what we expected, but it was just impressive that it could actually pull out those relationships.
Hi, my name's Kathy Garcia. I'm from the University of California, Irvine. So kind of on a similar vein, I was interested in predicting the surface variability of dissolved inorganic phosphate. This is an important biolimiting nutrient that has a different surface variation than, say, nitrate, for example. Using the database of GLODAP observations, we filtered it down to about 30,000 observations, and these happen to overlap with the remote sensing observations for the past 20 years or so. So looking at the variation in GLODAP observations on the right plot, sea surface temperature by itself is able to capture about 53% of the variation. But there's 2 challenges that I've sort of come across. One is that the variation in dissolved organic phosphate, most of the regions where it's going to be limiting are within the subtropical gyres, and the limitation to the measurements of common assays don't quite capture the variability of phosphate among different subtropical gyres. So using a high sensitivity database of phosphate observations and other neural network inputs beyond sea surface temperature, I'm able to capture about 73% of the variation in phosphate. So I'm able to overcome one of those first challenges of what's the variation in phosphate among very low concentration regions, but also temperature doesn't quite capture the variation in these really hot, high nutrient supply regions and upwelling systems. So by adding second predictors, including net primary productivity or sea surface salinity, we can try to capture this latitudinal variation, as well as variation among the different subtropical gyres and between different polar regions. And I just wanted to highlight that a lot of the support to kind of learn about remote sensing came from the OCB workshop. So thank you for providing the funding to go to that Cornell remote sensing course. Thank you.
Okay. For all the lightning session presenters, if you're in the middle of the crowd, it might be better if you just go ahead and move yourself to the outside. So next up we have Ellen Cliff.
Hello. So I'm Australian, so making this nice and international. So I've been looking at glacial ocean changes relative to the pre-industrial using the UVic and MOBI data. This is in the vein of today, though. We've been looking at oxygen disequilibrium. So here we've got a lot of the World Ocean Atlas 2018 and float data, and we can see seasonally there's disequilibrium, oversaturation in the summer, undersaturation in the winter, and especially in the Southern Ocean, undersaturation is very common. And we see this in our model, which is the blue line, and in the World Ocean data and float. So we use the MOBI model, and we split our model into the physical and biological components. I will go into the definitions of these more when you come and see my poster. It's a bit complicated, but it was cool to see the physical difference was not as big as we expected. The biological influence on oxygen as a disequilibrium and equilibrium was much more significant than the physical. So that's the basis of this. Thank you.
Okay. Next up, we have Fei Da.
Good afternoon, everyone. My name is Fei Da, and I'm a PhD student from Virginia Institute of Marine Science. And my poster today is focusing on the impacts of nitrogen from atmosphere and coastal ocean on the oxygen concentrations in the Chesapeake Bay. The figure on the left is the model I used to study the question. It's the Chestrom's ECB, which is described briefly by Margie earlier this afternoon. And I just removed the nutrient from river, atmosphere, or coastal ocean to do some sensitivity tests to look at their contribution on the oxygen concentration. One of our key findings are showed in the figure on the left, which is the change in oxygen due to the reduced nitrogen input in the summer. The first panel, so these figures are the transect along the main stem of the bay. On the left-hand side is the head of the bay, where the Susquehanna River coming into the bay, and on the right-hand side is the ocean. From this transect, the first panel, we removed the nitrogen from atmosphere, and for the second panel, we removed the nutrient from the river by 9%. This 9% is equal to the nitrogen from the atmosphere, so we have the same amount of nitrogen reduction for the first two cases, and we can see their impacts on oxygen are pretty similar. We can see about 0.3 increase in dissolved oxygen below the picnic line, which are shown by the dashed line. For the third panel, we removed the nitrogen from the coastal ocean. We can also see a similar increase in the dissolved oxygen, but have slightly larger increase closer to the open ocean, which is on the right-hand side of the transect. The figure on the right is looking at the problem from another view. We can see the relative contribution of each different nutrient on the bottom oxygen in the summer. The red is the impact from atmosphere, so we can see on the eastern part of the bay, nitrogen from atmosphere has largest impact on bottom dissolved oxygen. And the river has largest impact in the upper bay and in the larger tributaries showed in green, and closer to the open ocean, coastal nitrogen has largest impact. And in the deep trench in the middle of the Chesapeake Bay, the color tend to be white, means all three nutrient inputs, they have similar impact on the Chesapeake Bay dissolved oxygen. If you are interested more in the poster, please stop by my poster, and I'd like to discuss more details with you. Thank you.
Okay. Next up, we have Michael Fong.
Hi, my name is Michael Fong. I'm from the Scripps Institution of Oceanography. So I'd like to invite you to come talk to me at my poster, An Organic Source of Alkalinity in the Open Ocean. So I'll be presenting data from ghost ship repeat hydrography cruises where when we examine the consistency of high-quality CO2 measurements, there seems to be a suggestion that there is an unaccounted source of alkalinity in seawater. So here we have this term, the difference between the measured total alkalinity and alkalinity calculated from pH and dissolved inorganic carbon measurements using an equilibrium model that only includes inorganic acid-base species. So there's this suggestion that in the Pacific, Indian, as well as the Southern Ocean, there's an apparent excess alkalinity. And our hypothesis is that the source of this may be organic acid-base species. And while this hypothesis is not new, it's been proposed that this is important, particularly in coastal waters. These results are novel in that we've considered the various sources of biases that go into calculating this difference term. And they also suggest that this potential organic source of alkalinity is widespread in the open ocean, and that it also has a vertical and geographic distribution. When we look at this distribution of apparent excess alkalinity, so this AX, along a particular section, as well as the apparent oxygen utilization, we see that they seem to be correlated, particularly along certain isopycnals. So one of the hypotheses is that this source of excess alkalinity may be produced from the remineralization of organic matter. So if you'd like to learn more, please stop by my poster. Thank you.
Okay. Next up, we have Sophie Gill.
Thank you. Oh, thank you. Great. Hi, everyone. I'm Sophie Gill. I'm a PhD student from the University of Oxford in the UK, and I'm supervised by Ros Rickaby and Gideon Henderson. And I work on the calcification and growth response of coccolithophores to elevated ocean alkalinity. And the reason I'm interested in this is to do with the fact that I work as part of a greenhouse gas removal project. And one of the proposed schemes to remove greenhouse gases from the atmosphere, which is a key aim of the Paris Agreement to achieve negative emissions, is to enhance the natural weathering process, which is part of the long-term carbon cycle. So for the geologists among you, you may be familiar with this. And that process naturally draws down about 1 gigaton of carbon per year. So if we were able to accelerate that process and store the dissolved products in the ocean, we would increase alkalinity, and through shifts in the carbonate system, we would then draw down carbon dioxide. But there are two kind of major unknowns associated with this. Well, there are actually a lot of unknowns, but there are two that I'm trying to look at in my PhD. And the first is what the growth response is to increased alkalinity in major phytoplankton groups. And the second is what the calcification response of major calcifiers in the surface ocean is. And that's important because calcification releases CO2, so that could undo any net sequestration of CO2 that we're trying to achieve. So far, I have done batch culture experiments on Emiliania huxleyi and Gephyrochapsa oceanica, which are two of the major bloom-forming calcifiers in the surface ocean today. And I don't know if you can see this properly, but these are the flasks that I grew my coccolithophores in. And on the left in each picture is normal seawater condition, and then to the right we have gradually increasing alkalinity, and hopefully you can see that there's an increasing culture density in both species through my experiments, which is interesting, or at least I think is interesting. So if other people think it's interesting too, then definitely come and speak to me about more of my results at my poster. Cool, thank you.
Next up, we have Laura Holland.
Hi, my name is Laura Holland. I am a PhD candidate at University of Rhode Island, working with Bethany Jenkins. Oops. Oh, shoot. Yeah, I'm presenting all those slides. So my work focuses on two research cruises worth of samples, and we sought to look at the surface diatom communities within the Western Antarctic Peninsula. So, again, this is across two field seasons, 2016, which is in blue, and then 2014, which is in purple. And we tried to focus on three different questions. How do the diatom communities compare? Which environmental factors correlate with them? And what are the implications for biogeochemical cycling? And in particular, we were interested in seeing how dissolved iron might affect these communities, as there's this steep offshore to inshore gradient of low to high iron. Our DNA barcoding method showed us that the sampling sites between the years are actually quite similar, but where they differ is by sampling location. So the inshore samples where there's higher iron were different, significantly different than the offshore samples where there was lower iron. And to look at these differences in a little bit more detail, I did a co-occurrence analysis where I tried to identify co-occurring groups of different diatoms. And within the data, I found there were two main groups of diatoms that I saw. Fragileiopsis, so different species of Fragileiopsis, and then that was module 1 or group 1. And then module 2 is Pseudonychia and some small centrics like Phallaciosira and Catasters that form chains. And so these groups were strongly co-occurring in both cruises. And so on the bottom, we have a heat map showing how these two different groups are correlating with the different environmental traits that we found in the 2016 cruise specifically. I have the rest of the data on my poster. But for example, module 1 is positively correlated with dissolved iron and silica concentrations. Module 2 is negatively correlated with dissolved iron and silica concentrations. So from this, we can kind of think about how these different diatom groups are affecting biogeochemical cycling. And if you want to hear a little bit more about my speculations on that, please come to my poster. Thank you.
Next up, we have John Urban.
Hello, my name is John Irving from Florida State University, and today I'm just going to introduce a project we're beginning working on to try and quantify the sequestration time of carbon dioxide that's remineralized in the California current ecosystem. So POC can be vertically transported in the upper water column 3 ways: sinking, subduction, or active transport. This POC is remineralized into CO2 by bacteria or archaea, and they all have different impacts on, or each of these export pathways has a different depth at which they can be remineralized. So our goal is to see the sequestration time as a function of remineralization depth. To do this, we're doing very long Lagrangian modeling simulations by launching thousands of particles from the California current ecosystem. So for this first one, we're launching particles from 100 meters to 300 meters, and this is a 20-year simulation. We want to see where these particles reach the mixed layer, so then we can consider them no longer sequestered by the ocean. When looking at these particles, we can then look at the time distribution of how long it took for what percentages of particles to reach the mixed layer. Obviously, the more shallow particles, much higher percentages of them reach the mixed layer in the 20-year period, and in much quicker amounts of times. As we go deeper to 300 meters, a lot less particles reach the mixed layer, and it took longer for them to do so. Moving forward, we're going to combine this with the field data to actually compare this among the 3 export pathways, do deeper particle launches at 500 and 1,000 meters, and we actually want to see every particle get upwelled. So to do this, we'll probably have to do much longer simulations closer to 100 years. If you have more questions or anything to add, you can please come see my poster.
Okay. Next up, we have Thomas Kelly.
Good afternoon. I'm Tom Kelly from Florida State University. And today I want to show you something that has become kind of an exciting side project of mine. This is all data that was taken a couple years ago out in the California Current, just off of Point Conception. And what we wanted to do was start to compile multiple forms of productivity, because we realized that with our Lagrangian sample platform, we were in a perfect place to compare gross primary productivity with net primary productivity and NCP and export, and try to get a whole grasp on the carbon budget in these water parcels. So here, what I'm showing right now is a new method that we developed for calculating GPP from fast repetition rate fluorometry, which we were pretty excited about because this was a flow-through FRF that had really high temporal resolution, and we were able to understand the photophysiology, and we wondered, well, can we actually, in a realistic way, get that all the way to a practical GPP estimate for these water parcels? And what we found was we developed a pipeline, we developed a physiological model for the phytoplankton, and then taking a look at that in context with multiple other forms of productivity, we found that we did a pretty good job. So top left is a bar plot showing the rates of productivity for a whole bunch of different metrics, all in the same carbon units. And the two on the left are both GPP, one from instantaneous diurnal changes in O2 argon ratios to back out GPP. And the other one was from the FRF method that we developed. And we see that they compare really favorably, being completely independent metrics. Then the gray bar is NPP. You can think of this as traditional 14C 24-hour incubations. And that was about 40 to 60% of GPP. We thought we were on the right track there. The yellow is from dilution method. That would be phytoplankton growth. And then the blue, which is a small bar, and the green bar is new production from NO3 uptake, as well as net community production overall using a standard O2 argon method. And we were pretty excited about this because there are a lot of different assumptions that go into all of these methods, and the errors on them can be pretty high sometimes. But these are all very much independent metrics that we can now look forward to applying respiration from zooplankton as well as bacterial production, and look at the export production to try to get a closed carbon budget. Now, my poster is not on this, but it is on the same cruise across shore fluxes in a mesoscale filament going off the coast. So definitely stop by if you're interested, and thank you.
Next up, we have Heyori Lian.
Hello, everyone. I'm Hao Liang from Ocean University of China, and I'm a guest species student of in Hui. Today, I'd like to introduce about low molecular weight organic acids impacting on seawater acidification. In the Georgia Bay, we found these 3 different kinds of low molecular weight organic acids, and we found the formic acid and acetic acid have a strong correlationship. So it's indicated from similar source. And we investigate how low molecular weight organic acid impact on seawater acid by using the data from 2010 and 2017. The table is the major parameters of 2010 and 2010. And in the figure, we can see the pH, the measured pH is decreased in 7 years. And if we only consider the carbon dioxide system influence, we calculate the pH by using total alkalinity and DIC. The results is shown as the green square. We can see the results have an increased trend, which is opposite to the measured value. So we combine with the revised DOC value, which remove the low molecular weight organic acid in it, and we got the result shown as the blue triangle. We can see the value is lower than the green one. It's possibly because the rest of DOC contains some humic acid or other organic acid, maybe regarded as organic alkalinity will contribute to the pH decrease. And finally, we combine with all the measured parameters, include the low molecular weight organic acid, and got the result as the black triangle shows, which well fit as the measured value in 2017. So under the impact of low molecular weight organic acid, the pH decreased about 0.118 units. So it means the low molecular weight organic acid really has huge impact on seawater acidification, and we should not ignore it. Thank you.
Next up, we have Cynthia Michaud.
Hi, I'm Cynthia Michaud, and I'm from Moss Landing Marine Labs. And the data I've been working with, it comes from sediment traps at Station M, which is a long-term oceanographic station. The sediment traps I'm looking at are placed 600 meters above the bottom. The graph at the bottom is the whole time series of POC flux. The red represents the high flux periods. So those are defined as 1.5 times the standard deviation of the mean of the entire time series. So I'm looking at the high flux periods along with the events that occur right before and right after. So what I thought was going to happen was that during the high flux periods, you're going to have more cells sinking and a different community composition of phytoplankton. And looking at biominerals, you would see more biominerals sinking during high flux due to the ballasting of cells, making things more dense. So I used microscopy to identify phytoplankton cells and did biogenic silica analysis. Some results I found were that there were 2 distinct community compositions within the high and the low flux. The red is the low flux and the blue are the high flux. But when I look at the cell composition, I find that the relative cells are higher in the low flux events than in the high flux events. And then when looking at biominerals, the sinking particle silicification doesn't change between the high and the low fluxes, and the relative calcification decreases as you increase POC. And I'd be happy to go into more detail at my poster, and thank you.
Okay, next up we have Sarah.
Hi, my name is Sarah Nickford, and I'm working with Jamie Poulter at the University of Rhode Island Graduate School of Oceanography. So, western boundary current regions are areas of extreme CO2 flux from atmosphere to ocean, and this is amplified in the wintertime. However, observations are extremely limited. So on the bottom, that's February PCO2 from two databases, the SOCAT and LDEO. These databases are used to inform climatologies of CO2 flux and delta PCO2. One reason why these observations are so sparse is because it is hard to sample in the wintertime due to storms. So that motivates the use of autonomous surface vehicles such as Saildrone, which is pictured up on the left. And for the 2019 Gulf Stream mission, we had a Saildrone out sampling in the Gulf Stream, and it was equipped with PMEL's ASV CO2 sensor, which directly measures atmospheric PCO2 and ocean PCO2 to calculate the delta, which is an important component in calculating the flux. So on the top left, that is our cruise track, and plotted is the PCO2 measured by Saildrone. And comparing to the climatologies that are informed by the sparse observations, the Saildrone measurements suggest a 60% increase in the delta PCO2 in the boxed region around the Saildrone observations. And we also have observations from the Endeavour cruise, which followed the Saildrone mission at the end of February, in which it suggests a 30% increase from the climatology for delta PCO2. And increasing the delta PCO2 may lead to an upward revision of Western boundary current CO2 uptake. And if you want to see more data from the Saildrone mission, come stop by my poster. Thank you.
Okay, next up we have Frank.
Hi, I'm Frankie Pavia. I'm a grad student at Lamont Doherty. So I work a lot with in situ pumped, size fractionated particulate data. So this is collected with something like McLean pumps, particle sizes of 0.8 microns to 51 microns and greater than 51 microns. So most of what I work on is using method based on the 1D mass balance of the radioactive isotope thorium-230 to convert particulate measurements into particulate fluxes. So I recently did this with particular organic carbon in the eastern tropical South Pacific. And one thing we found was that our estimates were convergent across small particles, large particles, and sediment traps from nearby. And so this worked out pretty well, and we got really excited. We said, okay, now let's go do it with calcium carbonate. And when we tried to do it with calcium carbonate, we noticed that there was a difference between the estimates in small particles, large particles, and sediment traps, which necessarily implied some size fractionation in the pick-to-pock ratio of these particles between small, large, and what's collected by sediment traps. And so to investigate this, we put together a big database of all the size fractionated, small particulate, large particulate, inorganic carbon, and particular organic carbon measurements collected by in situ filtration since the 1970s up until pretty close to today. And so what I'll be talking about my poster is comparisons between these data. There's over 100 profiles of the surface most values of these data to satellite estimates of POC and PIC. And I think something very interesting that's come out of this is that the estimates of satellite POC from a monthly climatology reference to each data point consistently overestimates the surface most measurements from combined small and large particulate organic carbon by something like a factor of 2 to 4 for most of the data. So if you're interested in the regeneration length scales of POC from thorium-230 or calcium carbonate dissolution in the thermocline, or about how I should be using this satellite data to compare to my measurements, please come to my poster or find me and talk to me. Thank you.
Okay, next up we have Jessica.
Hi everyone, I'm Jessica Xu. I recently graduated from San Jose State University with my master's in computer science, and I emphasized in bioinformatics. So because of advancements in image-based methods, we are now able to quickly acquire more image data than we can manually categorize. And this amount of images just keeps growing. In our particular study, we have 17 categories of marine particles shown by these two tables. and we have hundreds of thousands of images. And so we are looking for a way to automate the process so that we wouldn't have to go through all of them. And so we wanted to build a machine learning model, an image classifier, to do this for us. However, the problem with that is that when you're building and training an image classifier from scratch, it takes an enormous amount of time and computing power. It takes a highly skilled person or team to build it. And one that we would build would probably be so specific to our project that it wouldn't be so robust, and also it wouldn't really help other people. And so instead, we've used transfer learning, which is taking an already developed model that's very powerful, and such as an image classifier that already knows how to classify cats and dogs, and teaching it or retraining it on our categories so that if we were to give it our images, it could give us a guess on what it thinks those images are. And with our experiments, which are still ongoing, we are just trying to observe what factors affect our accuracy. And so in this is one of our results for one of our data sets. We have 2 different illumination techniques and 3 data sets. So along the x-axis, it's the categories of marine particles ordered from smallest to largest training size, aka the amount of images that we had to teach the classifier. And on the bottom are precision and recall, which are just 2 metrics of accuracy. And one of the things that our results were able to show is that as the training size increases, or the number of images that we have increases, our accuracy significantly goes up. And so for other factors that might affect it, and also if you feel like this could help someone, please stop by my poster.
Next up, we have Patricia.
Thank you. Thank you all for being here. My name's Trisha Thibodeau. I'm a PhD student at VIMS with Debbie Steinberg, and I'm excited to talk to you all about my research regarding pteropods. Pteropods are beautiful open ocean snails whose wings allow them to swim through the ocean. This is an image on the left of the species Limacina helicina antarctica, which is an abundant pteropod in the Southern Ocean. And I actually took a video of these guys swimming on our most recent cruise in the Antarctic and posted it to my Twitter account, and it ended up going viral with over 18,000 likes and 300,000 views, which as a scientist studying snails is the best kind of outreach and support I can ask for. And you may have heard of pteropods because they're commonly associated with ocean acidification studies because their shells easily dissolve under more acidic conditions, and they use carbonate to build their shells, which is depleting under ocean acidification conditions. And we're concerned about this in the Antarctic in particular, but we don't have a very good understanding of pteropod ecology and physiology in the Southern Ocean. And this is important if we're trying to understand how they're going to be affected by climate change and ocean acidification. My research focuses on pteropods along the Western Antarctic Peninsula, and as part of my research, I've utilized a 25-year pteropod data set collected as part of the Palmer Antarctica Long-Term Ecological Research Program, as well as biogeochemical data to understand how long-term environmental controls are affecting pteropod abundance. And my analysis shows that pteropod, particularly Limacina abundance, is most strongly controlled by sea surface temperature and other environmental controls, including the El Nino Southern Oscillation. And this figure here shows that as sea surface temperature increases, Limacina abundance increases as well. I then wanted to see how these long-term environmental controls were affecting pteropod physiology. And so over the past 3 years, I've conducted shipboard experiments to look at the effects of projected sea surface temperature increases and food limitation on pteropod respiration and excretion. This figure in the middle shows that as sea surface temperature increases, we see an increase in pteropod respiration rates, particularly under low food conditions. We also see similar trends such as this in our excretion data, and you can come check out these results at my poster here at OCB. My research focusing on Antarctic pteropods will help us to better understand their contribution to carbon and nutrient cycling in a changing Southern Ocean. Thank you.
Next up, we have Laura.
Hello, my name is Laura Whitmore. I'm from the University of Southern Mississippi, and today I'm going to talk to you guys about freshwater in the Arctic Ocean. The reason we care about freshwater in the Arctic Ocean is because surface waters in the Arctic Ocean eventually leave the Arctic Ocean, where they have a role setting the pulse of Atlantic meridional overturning circulation. Because of this, it's important that we understand the inputs of freshwater, the storage of freshwater, and the export of freshwater out of the Arctic. I'm going to talk about one source. There's several. You can think of river discharge, sea ice melt, and what I'm going to talk about is the Pacific seawater entering the Arctic Ocean. You might say that's seawater, that's not freshwater, but it is less saline than Atlantic water, so it does have a freshwater component when we consider it in the Arctic Ocean. So I'm going to have you guys train your eyes on panel 1. What we're looking at here is a schematic of how we believe the distribution of waters in the vertical water column to look. What we're focusing on is that Pacific halocline, the gray area, and what's important for us to know is where is this Pacific water, and that's been difficult to tease out because we don't have good tracers for it. What we've been doing, and the reason we believe that this is the distribution, is using a nutrient tracer. This nutrient tracer optimizes on the differences in the nitrate to phosphate relationship in Atlantic water and Pacific water. It's inherently semi-conservative, so ideally we would be using conservative tracers to do this. So what I've done is apply gallium, which we expect to be more conservative, and tried to see what that distribution looks like. So if you look at panel 3 now, what we've determined from that is that the gallium method overpredicts the amount of Pacific water relative to this historic nutrient method that we've been using. So the blue line in panel 3 is the 1-to-1 relationship. So there is a big difference between these two methods. The next question we have is where is this difference occurring in the water column and possibly why? So panel 4 looks at where in the water column we see these differences. For the nutrient method, we see that it goes from 100% Pacific water near the surface to very rapidly 0% Pacific water. What that tells us is that there's very little mixing between the Pacific water above and the Atlantic water below. Alternatively, our gallium method tells us a slightly different story, and that is that there's more mixing than we had anticipated. I'm going to leave a big pause here and say, okay, what does this even mean? And that kind of takes me to this takeaway is, does the nutrient or gallium method more accurately represent the water column? This is again important for us to tease out as we start to consider how climate change influences these different freshwater sources to the Arctic and their distribution within. So this is a really exciting project, but it is not the project that I will be presenting tonight. So I do encourage you to check out my poster. I'm using barium as a tracer for a couple other things. But please stop by. I'm excited to talk to you guys about all things tracers, all things Arctic. So thank you.
Looking for a name. That's okay.
I'll say it. Hello, my name is Abigail Wyatt. I am at Princeton. I'm going to be talking a little bit about the exports trip. I went on the exports trip in September, and on the left here is a panel showing thorium flux. So I worked with Ken Bissler and Claudia Benitez Nelson. And what you can see here, as David already kind of described, was a little bit of the path that the sampling grid took. Here is the survey ship. So we were surveying around the Lagrangian float in 3 epochs, which is the 3 colors that you can see there. Blue is the first one, red is the bigger second one, and then green is the 3rd epoch. And so the size of the dots show the carbon flux, PC flux, and on the right, I'm looking at the same kind of thing in GFDL's Earth System Model 2.6. And so you can kind of get a sense of the variability in the model. If you can read the numbers, it's about, the bright red is about 10 millimoles of carbon to about the green, which is about 7, so maybe 3 is our variability there. And it matches the variability, although in the model we do see a mean bias that's a bit greater. We see a greater export in the model. And so one of the things I wanted to do, though, was to sample the model the way that we sampled the data. So using the same grid and the same time points, I looked into the model in the 2 years that we have daily data for. And I looked at that to see if we saw the same kind of story. So in the gray, this is kind of a complicated picture, and if you want to talk about it, you can come see my poster. But the gray is the model mean from the monthly data. And then we have 2 years of daily data, which are the orange and the blue. And then the green is the observational data. And this is SST as just a first look and to see what the model could do compared to what we were doing during the trip. And we do see similar levels of variability with the shading indicating spatial variability in the model. And then the green underway data from the Sally ride, you do see that variability as well. It's of a similar magnitude. So then one of the things, the reasons I wanted to use this model is because we do have this biogeochemical model inside the Earth system model that is moderately complex with 3 zooplankton groups here in blue, 3 phytoplankton groups in green, different labilities of dissolved nitrate, and then export that we can quantify directly. So one thing I was looking at is what's the variability of model export in different boxes inside the bottle centered on the exports track. And you can see that the variability increases in about July through September. You can see it widens. And we see similarly, when I looked at the biomass and phytoplankton and zooplankton, that we also see the shading representing the spatial variability, that variability increases from about July through September, which is during the period of the cruise. So I'm going to continue work in this and see what we can determine as the cause. And just as a first look, I've started looking at the limitation factors. And so you can see that when the large phytoplankton, the black line on the right is the same as the orange line on the left. For the large phytoplankton, we see that halfway through the year, the limitation changes from an iron limitation to a nitrate limitation. And so that's one of the things I'm going to start looking at further into the model. So if you'd like to discuss this, please come see my poster. Thank you.
I believe that concludes our lightning session presentations. So give one more round of applause to the presenters. And please feel free to stop by the poster session and see some amazing research.